But this is just an example, and I'm not going to go through the whole thing with you, but there is a mouse model for sickle cell anemia. And sickle cell anemia is where an individual, or a mouse in this case, would have a mutation in the beta globin gene. And that causes the red blood cells to not work properly. In this case, the scientists were able to take a cell from a sickle cell mouse, create iPS cells, exactly like I've told you already. But then in the lab, they were able to correct the mutation in those cells. So now it's no longer carrying a mutation in the beta globin gene. These genetically corrected cells, which are still iPS, so they're like embryonic stem cells, were then triggered to differentiate into blood cells, get re-injected into this mouse, and now the mouse recovers. So it gets a bone marrow transplant, but it gets its own bone marrow, which has been genetically corrected. So this is sort of the wave of the future, the futuristic dreams of what might happen. One other aspect of this that is similar, but doesn't necessarily involve disease, but might involve injury. The idea that you might have an injury in a tissue or an organ, you can grow cells, you can use the right combination of growth factors, trigger development of a new organ, and then implant that and recover. So I have one more little bit I want to talk about that has to do with this as well, and it's taking Yamanaka's idea and just tweaking it a bit. So this is a fibroblast where the 4 Yamanaka factors were used to make embryonic stem cell-like cells. Okay, so we're going to the very earliest stage of development, and then treating them with growth factors to differentiate them. And in this case, they're making neural stem cells. Scientists said, okay, well, we know we can go from a differentiated cell to a stem cell to a neuron. Can we bypass the stem cell part, the embryonic